Okay, now we're going to talk about one example for incomplete dominance dealing with a genetic disorder. Sickle cell disease is due to incomplete dominance. Big A people are normal. Big A little people have what we call sickle cell trait, and little a have sickle cell disease. Being normal is just like you and me. I don't think I've really ever known anyone with sickle cell disease. Sickle cell disease is very, very deadly. Their blood cells are sickle in shape, kind of like a crescent moon, and because of that, the blood cannot travel through the blood vessels properly because your blood vessels are circular in shape. The blood that has sickled up kind of gets caught and blocks blood, and so their blood isn't traveling through their body properly. There's oxygen deprivation that goes on, and it's very, very painful, and it can be quite deadly. And people that don't get adequate treatment will die. Sickle cell trait, they have a normal gene and they have a messed up gene, and due to the big A not being completely dominant over the little a, it's kind of like they have this middle ground going on. It's like they mix. So people with sickle cell trait will sometimes have their blood cells sickle up, and then they'll have an incident, and then they have to get medical treatment, and it's not as bad as the disease. Towards the bottom of the slide, you can see I have a couple of crosses there, and you could do Punnett squares if you wanted to practice doing that. I have a sickle cell trait person crossed with a sickle cell trait. Then I have a trait person with a disease person, and then a normal person with someone with sickle cell disease. The weird thing about having sickle cell is in areas where malaria is common. Malaria is a disease caused by a protist. It is spread by mosquitoes. Mosquitoes do not cause malaria. Malaria is actually caused by a tiny microscopic organism, and malaria is very deadly. Infants with little a or sickle cell disease will usually die. A lot of times because where there's malaria, it's kind of in these poorer countries and they don't have a lot of medical treatment. Infants that have sickle cell trait, or big A, little a, have a better chance of survival than actually normal people. People who have the trait, or big A, little a, have a protection against, big A, little a people have a protection against malaria. And again, that's a protist that uses the red blood cell during its life cycle, so it's a protist, a microscopic organism, causes malaria. And because with sickle cell, the blood cells are sickled up, that protist does not survive in these people's blood. So being sickle cell trait is actually better than being normal in areas where there is malaria. Normal people, or big A people, do not have sickle cell, but they'll die from malaria. Little a, little people die from the sickle cell disease. So an area where malaria is common, as many as 60% of the population will have sickle cell trait, big A, little a. Well, think about it. The big A, normal people, big A, they're dying from malaria. The little a people are dying from sickle cell, so the only ones you have left are the ones with the trait. They have this disease, but mildly, and they're not getting malaria. Win-win. Okay, on to blood types, which is an example of multiple alleles. There's 3 alleles that control your blood type, A, B, and O. And blood type is actually determined by the presence or absence of an antigen, which is a protein, on the red blood cells. So it's a protein that is sitting on your red blood cells. If you have the A antigen, you have A on the red blood cells, you would have antibody B. Antibodies fight things. B, you have a B antigen on your red blood cell, and you have antibody A. So you'd be fighting A. If you are O, it kind of means 0. You have no A, no B antigen on your red blood cells. You do not have any of them, but you have antibodies that will fight A and B blood. Each person has 2 of the 3, because you got one from mom and one from dad. And the thing with the blood, which I mentioned the other day, A and B are co-dominant to each other. A and B are both dominant over O. So here are possible phenotypes. There's type A, type B, type AB, type O. The genotypes, AA, AO, BB, BO, AB, because A and B are co-dominant to each other, and then the OO. And I did some crosses there, so that first one would be a type A person crossed with a B person. The second one is still a type A person, but they're homozygous for A, they're not the heterozygous, and they're crossed with a B person also. Blood typing can suggest someone is the parent, but you cannot prove paternity because millions of people have the same blood type. Blood type can prove that someone is not a parent. So if you look at the example I gave you, you have a kid who's O, you have a dad who's A, and a dad who's AB. That dad who's AB, there's no way he could have an O child. Okay? He cannot give an O to his kid. He's given an A to the kid, he's given a B to the kid. So there's no way the kid could be O, because O is just OO. An O kid has genotype OO. He got an O from mom and an O from dad. But that dad one, he could be AA, or he could possibly be AO. So the O then would have come from dad one. But we didn't necessarily prove that dad one was the father. Maybe there's another dad out there that the mom isn't saying. Maybe dad one has lots of money, and she's hoping that he bucks up and says, hey, I'm the father, and so she can get all kinds of money. So in this instance, we could suggest dad one's the father, but we can't say it for sure because there could be somebody else out there. You'd actually have to do what's called DNA testing to determine paternity. And usually when I talk about blood, students usually ask about the positive or negative, and that's just another antigen, another protein sitting on your blood cell, and either you have it or you don't. If you have the Rh protein, you are known as positive. If you don't have it, you are negative. So there's like the A positive, B positive, O positive, AB positive, and then the negative of each of those. Complications can arise when an Rh negative mother reproduces with an Rh positive man, and then they have a child who's Rh positive. The first time the mother has a child that's Rh positive, nothing really takes place, except at that point, because sometimes the mother and the child's blood can mix, that mother's negative. She now has positive blood in her from her child. She's going to produce an antibody. She's going to produce something that fights positive blood. It's not affecting her. It's just fighting that positive blood that got in her from her kid. Okay, everything's kosher. Well, then she gets pregnant a second time, and her second child's Rh positive. Their blood mixes. Now you have to remember that Rh negative mother has positive antibodies, has stuff that's going to fight positive blood. And if that mother and that second child's blood mix when she gives birth, that second child now has the antibodies from his mom, those antibodies that fight positive blood, and he has positive blood. It's going to cause issues with the child, and the child usually ends up with jaundice, which can be, it's not necessarily deadly, but there's some complications that can arise from that. Usually they try to do some testing of the mother and the father when a mother does get pregnant, and I think they can give her shots to prevent some of this from happening. So if you have any other questions about blood typing or Rh factor or anything like that, just put it on a note card and throw it in one of the question boxes that I have.